Hey, Jan.
Hey, how are you?
I'm good. How are you?
Hot. Oh, I'm tired of this.
Me too. I,
Just
I'm
crazy.
completely spoiled. I have a swimming pool,
Oh,
and every
we,
day I go. I have never been in my swimming pool so much.
yeah,
It's just so hot.
we go to Steiner almost every single.
And here we go. Welcome this afternoon to the Centers of Excellence in Hematology Meet the Expert seminar series. Really happy to have you join us, and want to thank Dr. Jan Christian for joining us today to present her talk. I'm going to take a minute to share a screen for our website so that I can guide you to some information. If you'll give me just 1 minute. Get it? There we go. So this is our website, which is the CCEH website. Wow, we'll get it one more time. And on this website, there's a couple things I want to draw your attention to. We have upcoming events like our seminar series that happens. We also have an opportunity for applying for funding. The funding that I'm going to describe to you here, I know this is going to start. I'm going to stop it, I hope. Is the type A and type B grants. Right now, the type B grants, which are these larger P&F program grants, are closed for applications. New applications will be accepted in February again, but type A grants are available all the time to apply for. And what they are is they're $5,000 to $12,000 worth of what we call core credits, where if you have a project that you need to use a particular core that's sponsored by the CCEHs, you can apply for some funding and get sort of deposited money at that core for you to do your work. So for example, I'll use the example of Utah. If you needed to do metabolomics or something like that, you could write a proposal, talk to the core director, who is James Cox, and he would write a letter of support, and you can apply for up to $12,000 worth of credits that are sort of stored at that core, and then you send your samples to them to run your samples. So I just want to remind people about that application process. And then the other thing I want to do is take you to this link that's called the service link, because you may not know what all the cores are that are available to you. And for example, how do I get my cells of interest? If you click on this link, what you'll find is several different little tabs here that you could hit on. So for example, primary human cells. If you click that link, you'll find that the Fred Hutch and Yale both have places that will help you with hematopoietic cell lineage isolation processes and getting those kinds of cells. Also, the Indiana Center of Excellence in Hematology has a flow and tissue cytometry core that can help you essentially figure out if they have some core tools there that might help you develop tools and also new methodologies for doing research. So if we clicked on this one, for example, this is their flow core. This is some imaging that they do, which essentially is a bone marrow, and they're doing imaging of a bunch of different cell types that are in the bone marrow. So I just wanted to draw your attention to that. That you might go to the website and see if there's a core that might be of interest to you. And let me just go back home again. The website is cceh.io. Very simple to find. And if you have any other further questions, you're welcome to reach out to me, and if I can help you in any way. I'm going to stop sharing my screen, and we're going to let Dr. Christian share her screen. I just want to take a minute to introduce her. She comes to us from the University of Utah. She originally came from Oregon Health Sciences, but she has studied iron homeostasis in the context of bone morphogenetic proteins and the regulation of development as well as hematopoiesis. And so this is an informal talk. I hope that you'll feel free to ask questions. You're welcome to populate them in the chat, but you're also welcome to interrupt and ask the questions. For now, I'll just ask people to mute themselves and then unmute themselves when they do have a question, if they would do that. And thank you very much, Jan, for your presentation today.
Okay, so can everyone hear me okay? Yeah, okay. So I feel like quite an imposter even showing this slide, but this is actually preaching to the choir just to say that iron homeostasis has to be tightly regulated to prevent disease, including anemia caused by iron deficiency or diseases of iron overload, such as hemochromatosis, where iron deposits accumulate in vital organs such as the liver. And to prevent these diseases, the body has a way of very tightly regulating iron. I've heard about iron regulation for the past, throughout my career. For the 20 years I was at OHSU, working with Carolyn Enns on all of her students' committees. And it seemed like we started off with something kind of simple, and over that 25 years, every new student and often every new student committee during the year, there'd be new players starting with transferrin, and then all of a sudden there's ferroportin, and then there's hemojuvelin, and et cetera, et cetera. So I'm going to try and keep this simple because I know your understanding is a lot better than mine. But just to keep things simple, I'll say that most of our iron is absorbed through the diet. We use it to build a number of tissues, including our red blood cells. And then iron is recycled through macrophages when the red blood cells die. And it's stored in a relatively innocuous form in the body bound to transferrin. So the major control points in keeping iron levels stable are a sole exporter called ferroportin that's expressed on iron exporting tissues such as the macrophages, the gut, and the liver. And then this hormone hepcidin, which is circulating, and its function is to bind to ferroportin and post-transcriptionally induce its degradation. So in response to, for example, high iron or inflammation, the liver senses this. It makes much more hepcidin to degrade ferroportin so that iron levels are normalized. And then by contrast, the opposite happens during erythropoietic drive. Whew, I'm onto a slide where I know something about BMPs. So work from a number of labs, and especially the Babbitt labs, has shown that the way that hepcidin expression is controlled is through the production of these BMPs, BMP2 and 6, by endothelial cells in the liver. It's secreted, it binds to classic BMP receptors together with this co-receptor hemojuvelin, and then activates phosphorylation of SMAD, which binds the hepcidin or HAMPS promoter to drive expression of hepcidin. And Jody Babbitt's lab a couple of years ago showed that most likely BMP2 and 6 are signaling as heterodimers rather than homodimers. And that data is kind of shown here. And what this is relative HAMP or hepcidin levels and liver iron stores in mice that are either genetically wild type, so they have high hepcidin, low iron, or there's a floxed allele so that you're knocking out either BMP2 or BMP6, specifically in these endothelial cells. And you can see there's a decrease in HAMP, an increase in iron. And the somewhat surprising finding is that you knock out both of these critical ligands. First of all, there's no further decrease. And this suggests that perhaps these two ligands are functioning together as a heterodimer rather than functioning redundantly as a homodimer. And then the other kind of surprising thing is that you never get rid of your HAMP completely. So there's probably some other BMP that's signaling. And my lab has, for most of my career, looked at how BMPs, different BMPs, not BMP2 and 6, how their activity is post-transcriptionally regulated, and also at the function of heterodimers during embryonic development. So Jody and I met at a meeting, and last year we were awarded an MPI grant, the goal of which for her lab is to ask what are the other BMPs that are contributing to regulation of hepcidin expression. And then in my lab, we're looking more specifically at how BMP2 and BMP6 are post-transcriptionally regulated, how their activity is regulated, and whether they're signaling as heterodimers rather than homodimers. So what I'm going to do today, we've been working on this for less than a year in my lab, so we don't have a ton of data about BMP2 and BMP6. And so I thought what I'd do is give kind of an overview of what we've learned about post-transcriptional regulation of other BMP ligands and about heterodimers. And then I'm going to intersperse in there some of the data for BMP6 that we've acquired to show how it's similar or different from these other ligands. Okay, so BMPs are all members of the TGF-beta growth factor family, signaling family. They play important roles both in embryonic development and in adult tissue homeostasis, for example, hematopoietic stem cells or iron homeostasis. And all of these proteins are made in the signal sending cells as inactive precursor proteins that dimerize through a disulfide bond near the carboxy terminus. And then they're cleaved by members of a family of proteases called proprotein convertases, and furin's probably the best known family member. At this motif where you've got arginine at the P1 and the P4 position, counting down from the cleavage site. And what this generates is the mature ligand, so that's what binds the receptor and initiates the signal. And then this piece called the prodomain. And the prodomain lacks any signaling activity itself, but we know it's important for ligand production. So it's kind of at the core, the prodomain for all TGF-beta family members is required to help them dimerize, to fold, and to get secreted outside of the cell. But then in addition, they have more specific functions that are specific for the ligand that they're hooked up with. So famously, TGF-beta prodomains keep the ligand in a latent or inactive form until it's activated by mechanical stress. Other prodomains can stabilize the ligand, they can help it to be secreted, and other functions. The other thing we know is that in many TGF-beta family members, in many BMPs, there's mutations in humans that, depending on the BMP we're talking about, either cause congenital birth defects, or in the case of BMP6, these mutations with the prodomain cause iron overload. And I'll come back to that a little bit later. But I think it's this finding that these mutations cause ligand dysfunction clearly demonstrates that they're important. They're just not just a throwaway piece. Okay, so over the past almost 25 years, we've been studying how one particular family member called BMP4, which is a really important developmental regulator, is proteolytically activated. And it turns out that it's actually cleaved twice. So there's an initial cleavage that occurs just adjacent to this ligand domain. And this generates this very transient but non-covalently complex between the ligand and the prodomain. And when the upstream cleavage site is cleaved, the whole thing falls apart, and now you've got the free ligand. And so we've been studying this over the years to ask, why do you do this complicated process? Are both of these cleavage sites required? And I'll kind of summarize and then show you a little bit of data. So first of all, we've introduced both in mice, in fruit flies, and then ectopically expressed in xenophus, ligands where you can't cleave the upstream site. So we've introduced a knock-in mutation, for example, in the mouse. And what happens is it stops at this intermediate complex. This thing can still be secreted. It can still bind to its receptor. It's not latent, but then it gets taken back up inside the cell and degraded in the lysosome. So the bottom line is that if you don't cleave the upstream site, you have less ligand made, and it signals only at a short range rather than a long range. And we've also found some evidence, both in mice and more so in Drosophila, that probably this upstream cleavage event is kind of a tissue-specific thing that will generate either this short-range, short-lived ligand, or a much longer range, longer-lived ligand. Okay, so we've also asked what happens if you fail to make this complex. So just to back up a minute, this first cleavage site is what we call an optimal furin motif. So in addition to the arginines at the P1 and P4 position, you've also got a basic residue at the P2 position, and furin really prefers this. If you only have the arginine at the P1 and P4, That's a minimal motif, and it's less well cleaved. And what we found is that's what drives furin initially cleaving this site and then cleaving this site. So we've generated, looked at this biochemically, and also generated a knock-in mouse where we've introduced this P2 lysine, and we call this the S2 site lysine mutation, or the BMP4 S2K allele. And what we can show biochemically is that you skip this complex formation and you just go straight to this long-lived, more active ligand. And we naively assumed that this might be a gain-of-function mutation, and the opposite turns out to be true. So it's actually a very severe hypomorphic allele. So this is a wild type embryo at about E11. Here's an S2K heterozygote, and then 2 S2K homozygotes. And I don't think you need to be a developmental biologist to see that these things are really abnormal. They have all sorts of defects: the eye, the jaw's missing, the heart's abnormal, vascular problems, all of which are incompatible with life, and all of which are associated with too little BMP activity. So we went on to use a BMP reporter mouse to show that this really is a loss of function, not a gain of function. And then we asked biochemically what's going on. So these now, they're just immunoblots of mouse. These are mouse lung lysates. These are mouse lung endothelial cells in culture. To look at the precursor and the prodomain in these lysates, we're using an epitope tag that we knocked in along with this wild type, or with the mutation or without. And you can see that a mouse heterozygous for just these tags relative to a mouse that has the tags and is also this S2K mutation, there's no difference in the levels of the precursor protein or of this cleaved prodomain fragment. But when you look at the ligand, you see something really different. So even in a heterozygote, the levels of cleaved ligand are really, really reduced, which is why we're having this profound loss of function phenotype. So just to summarize, if you fail to make this complex, the mouse is embryonic lethal and the mature ligand is turned over really rapidly. So what is this complex doing? And you'll see this throughout the rest of the talk, is that this complex really is transient. You can't see it if you just look at what comes out of a cell culture into the media. The only way you can see it is to do either a pulse chase or an in vitro cleavage assay. So what might it be doing? And one thing we know is that BMP4 prodomains bind to at least one ECM component that we're aware of, and probably more. And there's a lot of published literature showing that the same is true for the ligand. They bind to HSPGs, collagen, fibronectin, and in various contexts, these can either present the ligand to its receptor, they can stabilize it, they can destabilize it, but they're definitely functionally important. So this is our working model for what's going on. We're just suggesting that BMP4 precursor is trafficking through the secretory pathway with some ECM component already associated with the prodomain. And we've shown that BMP4 does not get cleaved in the Golgi. Both of these cleavage events happen as the ligand's being secreted. So what we suggest is that you get this first cleavage, you make the complex. This enables the prodomain to interact with things that are in the extracellular matrix, so that when the second cleavage happened and the ligand is released, it's basically passed off to some sort of an ECM component that stabilizes it, protects it from proteases. And then by contrast, if you cleave both of these at the same time, that prodomain fragment is still stable, but you don't have time to pass off the ligand and it's rapidly degraded. Okay, so the question then is BMP2 cleaved in exactly the same way? And I just want to introduce that developmentally, there's 2 classes of BMP ligands that we consider as really important for regulating early embryonic development. Class 1 consists of BMP2 and 4, and then class 2 is 5, 6, 7, and 8. And if you look at the sequence, the Prodomains sequence of these class 1 ligands across species, what you see is that we have this 100% conservation of an optimal furin motif at the S1 site, and then this minimal motif downstream of that. And so we had every reason to believe that BMP2 is going to be cleaved exactly like BMP4. And it turns out that that actually is probably not the case. So what this experiment is we're making recombinant radio-labeled precursor proteins just using reticulocyte lysates. Okay, and then we incubate this radio-labeled precursor with recombinant furin in a test tube, and at various times after adding the furin, we take out aliquots and run it on a gel and use fluorography to look at the cleavage product. So for BMP4, here's the precursor protein. It goes away over time. What you're seeing here is just a prodomain band that's been cleaved only at this S1 site. And then over time that disappears and it's converted into this S2 cleaved prodomain. So that's really the evidence that this cleavage is sequential. But with BMP2, we see something completely different. What we see, no matter, and Pooja is the postdoc who's been working on this, and what she sees very reliably and reproducibly is that at all time points, she only sees the S2 cleaved prodomain. So she's also looked at mutant forms. So for example, here's an S1G mutant where you can only cleave the S2 and not the S1. That gets cleaved just fine. This is the opposite mutant where you can't cleave the upstream site, but you can cleave this optimal furin motif. And surprisingly, what she sees is very, very little S1 cleaved prodomain product. And the precursor, again, doesn't disappear over time of incubation. So it almost seems like you need to cleave the S2 site in order to cleave the S1 site. So
Hey,
just, yeah.
Will you go back? Oh, you now have to go back one slide. Are there amino acids that flank that domain in BMP2 that make it different, that are regulating that?
Probably not. So furin really, it recognizes not just these 4 in its motif, it really recognizes up to P6. And if you look at BMP2 relative to, for example, BMP4, right, you would almost assume it's going to be cleaved better. It's got these basic residues. And with regard to flanking residues, I'm not going to talk about this, but this histidine at the S2 site serves as a pH sensor and is always also really important. So there's really nothing we can see sequence-wise that's different.
Okay, thanks.
Yeah. Okay, so what's going on? So BMP4 does a sequential cleavage. BMP2, either both of these sites are cleaved sequentially, or you're cleaving first the S2 site and then the S1 site. And we have a lot more work to do to figure out what that means if this is what's happening. I will say, so what Pooja plans to do is to just repeat this entire analysis, but rather than looking at prodomain cleavage, she's going to look at the ligand. She's going to optimize the gel to look at the ligand. So she'll be able to see if the S2 site is cleaved first and then the S1 site. So one other piece of data, though, is just looking at what happens in vivo in HEK cells. So they're just transfected with wild type or these cleavage mutant forms of BMP2. That's a typo, that's not a 4. And now she's looking on immunoblots at the ligand fragment. So BMP2 is cleaved at both sites. The S1G mutants cleaved only at the S2 site. And again, this, if you make a mutation at the S2 site, nothing gets cleaved. So I started out thinking this is artifact, and I have to admit that it was while I was putting together this talk that I remembered work from our lab and from another lab that's looking at cleavage of the Drosophila ortholog of BMP2 and 4. So there's only one DPP, one fly. 2 and 4 are basically duplicated genes. So fly-like vertebrates has an S1 motif that's an optimal. It has this S2 motif. It's also optimal rather than minimal. And then what work from the Shimmy lab here showed is that there's a third cleavage site internally here. And what they went on to show is that you actually have two functional ligands in fly. The short ligand that's cleaved at S1, longer ligand that's cleaved at S3, and they're both functional in vivo. But what they also showed is that unlike BMP4, where if we make a mutation so that you can't cleave the S2 site, S1's cleaved just fine, it's unstable, it's short range, but honestly in a mouse, this is a really mild hypomorph. So this cleavage is not critical. In DPP, by contrast, if you make a mutation where you can't cleave the S2 site and you look in the wing disc by immunoblot, you find that the ligand is not generated. It's just not cleaved at that S1 site. And because of this, if you make a mutation in DPP in the wing, you get no wing. You can completely rescue this with wild type, but not this S2 mutant form of DPP. And what I had forgotten, it was a little bit, yes. So our lab in collaboration with Marcel Worley did exactly the same thing at the same time, not knowing that both labs were doing this. And we found identical to the Shimmy lab in the wing disc, no ligand on the immunoblot, can't rescue activity in the wing. But if you look in another tissue, which is the fly gut, now this ligand that has a mutation at the S2 site, it's cleaved, it generates this ligand just fine, and it's fully functional in the fly gut. So there's clearly something going on with tissue-specific use of this upstream S2 site. And I think it potentially might be really interesting that BMP2 seems to be functioning more like DPP than BMP4 in terms of looking what it's doing in different tissues, including in liver endothelial cells, obviously. Okay, so briefly, I just want to, I've introduced type 1 and type 2 BMPs. BMP7 is an example of a type 2 BMP. It's the only one that's been looked at in terms of proteolytic maturation. And what we know is that BMP7 is cleaved at a single site. It forms this really stable non-covalent interaction or complex with the prodomain, still associated with the cleaved ligand domain. And it's been shown biochemically that the place where the prodomain interacts is the same place where the ligand binds to a type 2 receptor. So through a lot of biochemical modeling from the Sengel group, they've come up with this kind of model, which has been partially but not fully tested in vivo. It's mainly biochemical modeling. But in this cartoon here, the blue is the prodomain, the red is the ligand. And when you have this complex soluble in solution, it can interact with the type 2 receptor, and that displaces the prodomain. The other ligand binds, displaces the prodomain, and the thing can signal just fine. But when you're in a tissue that is expressing fibrillin microfibrils, the prodomain binds to those microfibrils, and it puts it in kind of a closed conformation where the prodomain is obscuring the receptor binding sites. And so it's keeping it more or less in a latent complex. So we've just started to look at cleavage of BMP6 and whether it does the same thing. We don't have a lot of progress there yet, but I want to introduce these human prodomain mutations that I mentioned earlier that are associated with iron overload in humans. And the reason for that introduction is that these green residues here, this is BMP6 and 7 aligned. So in BMP7, these green residues are the part of the prodomain that interacts with the ligand and can interfere with receptor binding. And interestingly, 2 of these point mutations in humans are within this small domain. Another 2 are in this stretch of Q-rich residues. And this insert here is unique not only to BMP6, but it's unique only to mammalian BMP6. Even lower vertebrates don't have it. And then this last one is further downstream in the prodomain in a region that's thought to modeling based on other TGF-beta family members. It's like a hydrophobic rich region that is important for interactions. So we have started to test whether these mutations in the prodomain interfere with BMP6 activity. And just to introduce the assay, this is a Xenopus embryo assay. During gastrulation, BMPs are really strongly expressed in the ventral and lateral regions of the embryo. You don't even need to know what those are to understand this. In the opposite side of the embryo, there's no BMP, but it's really enriched in BMP inhibitors. And as a result, phosphosmad just isn't even active in these cells. So what we do is we inject RNA encoding, or PUJA injects RNA encoding wild type or point mutant precursors near the dorsal side of the embryo, dissects out this dorsal region, and then does a immunoblot for phosphosmad. And what she sees is this 92S mutation leads to about a 50% reduction in pSmad activity, whereas this residue immediately next door has absolutely no effect. And the Q111E mutation causes, in general, even a more severe reduction. Now, we've only done this twice, so we don't have error bars, but it seems to be reproducible. So in Xenopus embryos, 2 of these mutations do interfere with activity, 2 really do not. We've also tested this in cells. So in this experiment, and it's a little artificial, and Jody and I have been talking about ways to try and optimize this, and Allison as well. So we're expressing our wild type or mutant precursors in HEC293 cells, collecting media after 24 hours, putting it on HEP3B cells, and then blotting for pSmad. And in this case, what we see is, again, these same 2 point mutant forms show a severe reduction, and it's even worse in the cell lines in the ability to activate phosphosmad1. And the reason for that, if we look at this media and look at how much ligand is in it, is that ligand generated from these point mutant precursors is at much lower levels, barely detectable, in fact, in the case of this Q11E mutation here. So obviously, this is a little bit of an artificial system. The cells in vivo that are actually making this BMP6 are liver endothelial cells. And so Allison and Jody and I are talking a lot about whether we want to use, for example, HUVEC cells, or possibly what would be even better is if we could use primary liver endothelial cells as the cells that are expressing the precursor protein, since that kind of seems to be where the problem is rather than reception. Okay, so I'm gonna transition to heterodimers now. So class 1 and class 2 BMPs can function either as heterodimers or as homodimers, or as heterodimers, where you have one ligand and one class 2. And the example here is BMP4 and BMP7, but basically the same is true for any other class 1, class 2. So BMP26, BMP27, 47, 46, etc. And what's been known for a very long time is that if you just put a certain amount of each homodimer into your favorite assay, it generates a certain amount of activity, whereas a heterodimer generates much more. And this can range from 10-fold more to like 100-fold more. So they're clearly, cells are more sensitive to the heterodimer. And so we wanted to know, do heterodimers form in vivo, and are they the endogenous signaling ligand? So this is an assay where we're looking at whether they form in vivo, and we're just injecting Xenopus embryos with precursor protein for 4, 7, or both together. And the embryo then processes the protein, it secretes the ligand and prodomain fragments into this blastocele that we can then suck up and look at the ligand on immunoblots. So on non-reducing gels, you can see that BMP4 homodimers run here, 7 homodimers run here, and when we co-inject, they preferentially form the heterodimer over either homodimer. So this is, again, a little bit of an artificial system. We don't always get exactly the same amount of each RNA injected. In this case, you can see that there's more 7 than 4. And if you were to look under this arrow, you can see that there's a little bit of BMP7 homodimers. So the point is that if these are co-expressed, they'll first make a heterodimer, and then if anything's left over, they'll make a homodimer. And we wanted to ask what's going on in vivo. So this is kind of a model where you can, and this was very controversial when we first published this. There's groups of people who are positive that only homodimers form, and that this heterodimer thing is an artifact of people making them and putting them on cells. So the way we were thinking about this is we generated a knock-in allele in BMP7. so that we introduced at the cleavage site this glycine residue, so furin can't recognize it. And the idea is that if you only make homodimers in vivo, then the only thing you're going to take out with this cleavage mutant is these homodimers, and therefore the phenotype of these mice is going to be just identical to that of BMP7 nulls, which live until birth and then die of kidney disease. Okay, but by contrast, if you're making heterodimers, then you're going to make these complexes with BMP2 and BMP4. You're going to take out these really active signaling molecules, and the phenotype's going to be a lot worse. So, and that is in fact exactly what we saw. So these are just mouse embryos, again, at E10.5 and 11.5 BMP7 nulls. Look exactly like these embryos where we've knocked in just an epitope tag, but nothing else, or wild type litter mates. Whereas homozygosity for this cleavage mutant allele gives an embryo that's smaller and has some serious heart defects by E10 and is basically runted and resorbing a day later. So I'm going to skip these next few slides in the interest of time because they're probably of more interest to developmental people. And then finally, we've asked whether we can detect these heterodimers, endogenous heterodimers. And so again, we have this flag tag knocked into the BMP7 allele. We collect lysates for embryos heterozygous for the flag allele in a wild type or an RG context. IP with flag to pull down the BMP7 precursor and then blot with BMP4. And what we can see is that when we do that, we pull down both the BMP4 precursor and the BMP4 cleaved ligand. So what the embryo is making is this uncleavable form of BMP7 as a heterodimer with BMP4, and this is cleaved, but it still can't signal. Okay, so BMP4 and 7 preferentially form heterodimers when co-expressed. BMP7 seems to function primarily as a heterodimer with 2 or 4. And then the question is it the prodomain or the ligand that's driving formation of these heterodimers? And we've looked at this in 2 ways. One of the ways is just to generate recombinant BMP precursor or ligand in bacteria. And then we refold and dimerize either BMP4 alone, BMP7 alone, or equal amounts of 4 plus 7 in vitro, just using published protocols. And what we find is if we do this with the precursor protein, we see BMP4 precursor homodimers migrating here, 7 homodimers migrating here, and the heterodimers migrating higher. And in this case, again, we're not seeing either of any homodimer precursor. But now if we repeat this, but we take away the prodomains and we're only refolding the ligand domains, we see something really different. So now you're making equal amounts of each homodimer together with some heterodimer. So I think even in a cell-free system, it's the prodomain that's making the decision that you're going to be a heterodimer and not the homodimer. Okay, so we've also done this in vivo in Xenophus embryos for both BMP4/7 and now for 2/6 as well. So this assay is a little bit complicated, but what we're doing is injecting RNA encoding 4 precursors, 7 precursor, or both together with a lineage tracer. And then a day later in development, we're looking at where the lineage tracer is by staining for beta-gal, which is these red dots here. And then we look for a BMP target gene by in situ. And the embryos show a range of staining from a 0+, which is nothing, a 1+, where the signals completely overlap, 2+, where you've got blue XPRA staining outside of the lineage tracer, and then you've got staining across the entire embryo. And to make this easier, what others and we have shown is that at this dose of injected RNA of BMP7, it doesn't signal in this assay. It's a 0+, whereas BMP4 signals primarily at a 2+ level. And so now if you inject half a dose of each of these together, you're not getting any contribution from BMP7, but now even a half dose of BMP4 and BMP7 gives you an even stronger signaling profile than the P4 alone. Okay, So what we did is we generated chimeric precursors. So initially what we have is a BMP4, a cDNA in which the BMP4 domains fuse to BMP7 ligand. And we make RNA and we inject it and compare it. And looking biochemically, so again, here we're injecting B4, BMP7, BMP47, here's homodimers, here's the heterodimer. When you try to generate the BMP7 ligand from the 4 protein, that seems to work just fine. You're getting ligand produced that's migrating at the right position at the gel. Now, what was surprising is if you look at activity in the embryo that's generated by BMP signal ligand from its native precursor, it does signal. But now when you generate the exact same ligand in the context of the BMP4 prodomain, it has really high activity. It looks just like BMP4. So that was a surprise. And then if we repeat this, but now we're asking, can you make heterodimers in the context BMP4 prodomain alone? And the answer is yes. Here's your heterodimers, and those heterodimers are more active than either homodimer. Okay, so we've done the opposite then. Now we have BMP7 prodomain, BMP4 ligand. Here's BMP4 homodimers made with its own prodomain, made with the 7 prodomain. It looks okay. It's kind of migrating like it's not entirely folded, consistent with that. Here's activity BMP4 from its own prodomain, significant loss of activity if you're driving it from the 7 prodomain. And it's even worse if you try and make heterodimers. So heterodimers should be migrated here. There are none. These are probably misfolded heterodimers. You make some BMP7 homodimer, but these are completely inactive. So just a summary, the BMP7 prodomain can't make a stable BMP4 ligand that can signal in vivo. But the opposite is that the P4 prodomain can make a BMP7 ligand that can signal in a context where it normally is completely inactive. And then the same is true for heterodimers. So what's going on? And again, we're still doing a little bit of hand waving, but we think that the same thing goes on with heterodimers, goes on with BMP4 homodimers, where you're trafficking through the secretory pathway some chaperone protein that can integrate into the extracellular matrix down to the prodomain, passing off the ligand to something that enables it to signal and to stay stable. The same is true for these heterodimers, and the same is probably true for BMP7 homodimers, where their own prodomain can't present them to the receptor in this tissue in a way that they can signal. The BMP4 prodomain can do that. Okay, so briefly, we've done the same with 2 and 6 now, and I'll just tell you, this is again by Pooja, but it was started by Allison Fisher when she visited my lab last summer. So now rather this in C2 assay, we're using a more quantitative assay where we isolate this ectodermal explant and look at expression of the gene XBROW and renamed TBXT by PR. So BMP2 induces high levels of TBXT. BMP6 is really inactive, so it looks like every class 1 BMP can't signal in the ectoderm, probably because of its prodomain. If you hook up the BMP6 prodomain to 2, you get a significant loss of activity. If you hook up the BMP2 prodomain to 6, you get significant gain of activity. So BMP2 and 6 in Xenopus at least are functioning similar to 4 and 7. That's quantitated here. Okay, so this we have only repeated twice. It's reproducible though. And this again is just expressing wild type or chimeric precursor in HEC cells, treating the Hep B cells, immunoblocker, PSMAD. And I just want to show this because it's really different than what you see in the embryo. Here, both BMP2 and BMP6 are stimulating significant PSMAD induction, which I guess we know from in vivo hepatocytes are very responsive to both of these ligands. But now if you express 2 in the context of the 6 ligand, no activity. And the same is true of 6 context of the 2 ligand. And I think just reinforces that we really need to be using the per cell lines to do these experiments that are more recapitulating what's going on in vivo. Okay, final few minutes here. I want to go back to 4 and 7 and just show you some unpublished data. So it known that similar to 6, there is humans that have heterozygous point mutations within the prodomain of BMP4 because this is a developmental regulator. They tend to cause congenital birth defects rather than iron overload. So the question, given that we know that 7 is signaling primarily as a heterodimer with 2 or 4, we wanted to know, are these mutations taking out heterodimer activity, homodimer activity, or both? And I think this is just some crystallography work from the Hyben lab and also the Spring lab, where they've crystallized for the first time precursors rather than just the domain for both activin and TGF-beta. And what they find in both cases, if you look at this precursor monomer, the gray is the Ligand and the red. The green is the prodomain, and you can see that there's parts of this prodomain on this monomer that are very tightly associated with the ligand that it was originally covalently attached to. But now when you make a dimer, what you can see looking at the same ligand is that a lot of the prodomain that's from the opposite monomer is now interacting with this ligand domain, perhaps helping it to fold, perhaps enhance activity. So if you think about a 4-7 heterodimer, the same would be true, and it's possible that these mutations are so detrimental because they're interfering with heterodimers, sorry. So this is one mutation that we're particularly interested in. It takes place in this residue motif in the prodomain SGE. And there's 2 cleared mutations here. One is a mutation of the this E residue to a G. We call that E3G. And the other is a mutation of the serine residue to a cysteine, or S91C. And these lead to defects such as cleft lip and pet, smaller absent eyes, renal hypodysplasia, brain malformations. And the reason we're so interested in is that we're shown that this serine is actually phosphorylated, and it's phosphorylated by a secretory pathway kinase called FAM20C that phosphorylates at the motif XE. So mutating either of these should prevent that phosphorylation event, but we have no idea what it does. So we've modeled this in Xenophus and mice. This is again that TBX-T assay where we're looking at BMP or homodimers, wild type BMP4, strong inducer. Either of these mutations leads to a considerable reduction in ligand activity. But when you look at heterodimers, all 3 of these prodomain mutations seem to have no effect whatsoever. So this was a little surprising to us, but now this phosphorylation event, it's to primarily be affecting homodimers. So given this, I think what was even more surprising is that we generated a mutation in mice at ASS91C together with an HA tag and a call out to Crystal and Kyle in our course. And these mice are embryonic lethal. Mice homozygous for this mutation are embryonic lethal. And it occurs early. They're abnormal by E10. By E11, they're grossly abnormal and resorbing. We've all isolated MEFs from heterozygous mice and then analyzed levels of ligand and phosphosmad1 in these MEFs. And this was just the first experiment. We've reproduced this. This is a more drastic reduction than what we've seen subsequently. But these are out of order because basically we did the blot before we genotyped. And what's really obvious is that there's this reduction in phosphosmad1 levels when mice are heterozygous for this mutation. So I'm just going to finish by saying this brings up a lot of questions. So our analysis of a BMP7 cleavage mouse says that heterodimers, BMP7, a lot of the activity embryo generated by BMP7 is coming from hair dimers. And yet the fact these S91C mice show a very similar early lethality brings up the question of whether BMP4 homodimers are also contributing significantly to early embryonic development. So I am going to finish there and just acknowledge the people who did the work. Hyung-suk, the cleavage mutant mouse, Pooja's working on BMP26, Mary presenting her poster. She spearheaded the S91C project, and Josh has taken it over, and previous members though. And I'd be happy to take any other questions.
Great. Thank you, Jan, very much for the presentation. Does anybody want to ask questions or have a question they want to talk about? Anna says, nice talk, Jan.
Thank you. It's a lot of data overload for people who work on iron homeostasis, a lot of mutant mice.
The biochemistry is very surprising.
It's very surprising. And I have to say, I don't, unfortunately, not going to get a definitive answer on what the heck the prodomain is doing with the matrix. That's what's going on in my career.
Yeah, I was trying to figure out if there's something happening in the secretory pathway that's kind of clogging it or something like that, especially if there's some sort of interaction with an extracellular matrix that's going out.
Well, you know, TGF-beta itself, which is the best studied prodomain in history,
Yeah.
it actually has a chaperone that binds covalently to its prodomain. And it needs that to get out of the secretory pathway, and it anchors it in the matrix. So I think this idea that that might be what's happening, there's a lot of precedent for it. But knowing what that factor is and how it's functioning is proven to be much more difficult.
Trying to think about a screen. If there's a screen you could run.
Well, we've definitely talked about proteomic screens. I think they'd have to be done in vivo because this whole process is so intricately tied to folding.
Yeah.
That I think you can't just express the prodomain in vitro and say what's it binding to.
Are there questions or comments? You're welcome to unmute yourselves. You're welcome to, excuse me, come off your video.
Okay, well, if there aren't any questions, I'm going to run off then and dissect a few more mice before my next seminar. So thanks everyone for your attention.
And thanks, Jan. Really appreciate it. And thanks, everybody. And please share with your friends that this recording will be made available on the website. Hi, Jody.
Hi, thanks. Beautiful talk, Jan. I learned something new every time I hear you talk. So.
Oh, thank you.
Yeah. Okay. We'll let you run off, Jan. And thanks, everybody.
Okay.
Okay.
Bye.
Bye.